Hello, my name is Jenay, and I'll be sharing about my biology role model project on Dr. Victor Ambros. He's a molecular and developmental biologist who changed how we understand gene regulation. Victor Ambros was born in 1953 in Hanover, New Hampshire, and grew up on a small dairy farm in Hartland, Vermont. As one of the 8 children, he learned to be curious and resourceful. He loved fixing tools and exploring nature, but that sparked his interest in early science. By around age 9, he was already fascinated with how living things worked. He first considered astronomy, but later discovered his passion for genetics and molecular biology. Ambros studied biology at MIT, earning both his bachelor's and PhD under Nobel laureate David Baltimore, and at that time he was studying how viruses replicate. After his PhD, he joined Robert Horowitz's lab to study development in C. elegans, a tiny roundworm often used in genetics. He later became professor at Harvard, then Dartmouth, and today he's a researcher at the University of Massachusetts Chan Medical School, where he continues studying gene regulation. Outside the lab, Dr. Ambros is known for his curiosity. He enjoys tinkering and repairing guitars with his wife, a hobby that reflects hands-on mindset he developed on the farm. He often says that every day in the lab is an adventure, showing how much he values discovery and the process of doing science. In this picture, his wife watches him as he fixes one of the guitars. I admire Dr. Victor Ambros because he discovered something revolutionary, microRNAs, which was not an accident, but through patience and curiosity. He paid attention to the small details that others might have overlooked, and that curiosity led to one of the most important discoveries in molecular biology. I also connect with him because of his humble and persistent approach to science. He reminds me that determination, curiosity, and staying open-minded can lead to incredible breakthroughs. Victor Ambros works in molecular and developmental biology, focusing on gene regulation and how genes are turned on or off after transcription. He studies microRNAs or miRNAs, which are basically small RNA molecules that don't make a proteins, but instead regulate which genes are expressed in a cell. His guiding question, which he focuses on, is how do microRNAs control gene expression and influence development and disease? In this picture, you can see the C. elegans, which was his main focus of research. One of Ambros' recent paper, published in 2024 in PNAS, is titled Modeling Neurodevelopmental Disorder Associated in HAGO1 Mutation in C. elegans, Argnot ALG1. In this study, his team recreated the human disease mutations found in the ARGNOT1 gene, a key part of the microRNA machinery, inside the worm, version of the gene called ALG1. They wanted to see how these mutations would affect microRNA regulation and development. Here is figure 1, and we will jump in the separate panels now. So in the top left, or in the first panel, we see that the sequence alignment of ARGNOT proteins from humans and worms. The labeled positions like F180, G199, B254, and others mark where the mutation occurs. These are conserved amino acids shared between human and worms. In the next panel, we can see that the protein structure diagram shows ARGNOT's domains, N, L1, PAZ, L2, etc., with arrows pointing to where each mutation is located. These positions are near regions important for microRNA binding and function. Moving on to panel 3, in the graphs show the adult survival rates at 20 degrees Celsius and 25 degrees Celsius in worms. At the first top left graph, we can see that the gray bars are normal worms and the red bars are mutants. Notice how the survival drops dramatically for the 8751L mutants, especially at 25 degrees Celsius. This means mutation disrupts the ALG function. Moving on to the last panel, we can see the fluorescence images that show expression of a gene reporter, the COL19 gene. In the top image, labeled WT pattern, the green fluorescence marks show normal adult cells. In the middle and the bottom images show mutants, and the fluorescence is dim or missing, showing that the development timing is disrupted. Together, figure 1 shows the recreation of human AGO1 mutation in worm, and it disrupts the microRNA pathway, leading to development and fertility problems. This connects molecular defects in ARGNOT to broader development consequences, both in worms and potentially in human. Dr. Ambros' most famous discovery was in 1993 when he and his colleagues identified the Lin4, the first microRNA. They showed that this small RNA binds to another gene's messenger RNA and blocks it from being translated into the protein. This finding completely changed how we think about gene regulation, and it proved that RNA molecules themselves can control gene activity. Since then, scientists have found that thousands of microRNAs across plants, animals, and humans, and they're now known to influence development, cell differentiation, aging, and even cancer. Ambros' lab now studies how microRNAs are made and how they recognize their targets and how they might be used in RNA-based therapies. In 2024, Victor Ambros shared the Nobel Prize in Physiology and Medicine with Gary Ruckman for the discovery of microRNAs and their role in post-transcriptional gene regulation. Victor Ambros changed biology forever by discovering microRNAs. His work revealed an entirely new layer of gene regulation and opened the door to new medical possibilities. What inspires me most is how his breakthroughs came from curiosity and persistence. In the future, his research could lead to new RNA-based treatments and better understanding of diseases linked to gene regulation. Dr. Ambros reminds me that even the smallest discoveries can change the world of science. Here are the sources. And thanks for watching.